Hello everyone, my name's Phil Bourne. I'm actually a professor in the Skaggs School of Pharmacy. I do early stage drug discovery. I'm also the Associate Vice Chancellor for Innovation and Industrial Alliances here on campus. And that's a new role for me. It's something since this class was given last year. And I have quite a different perspective on things than I had before as a result of that. And I'm not going to touch on too much of that today, but it's clearly something that is nagging at me right now in terms of how we get things out of the university into clinical trials and of course beyond. I think there's some huge challenges there, and I've heard a bit about, you touched on that already, and perhaps we could talk about that at the end. Okay, so I'm going to focus on the idea of proteomics and genomics and what that brings and how that's perturbing the drug discovery process. And I would say that you've kind of heard a bit of this. Let me just cast it in my own way by saying it's the best of times, and in some ways it's the worst of times. And I love this. This is the opening paragraph from Charles Dickens' book, and it's so apropos for drug discovery. It's unbelievable. It was the best of times, it was the worst of times, it was the age of wisdom. And I think genomics and proteomics is kind of giving us the age of wisdom, but there's also the age of foolishness in how we've so far been able to take a lot of that forward because we haven't changed the way we do things in large part in the drug discovery process, even in light of this new data and these new capabilities, in my opinion. And I'll give you some illustrations of why I think that's breaking down. And I could go on to this with the winter of despair and so on. But here's the sort of best of times, right? We are in this omics revolution. It started in about 1990 with the Human Genome Project, which took quite a long time to actually bear fruit after it was finished in 2000. But there's no question, and there's no doubt, and you asked the question about personalized medicine, that as we move down, we're at whatever it is now, $3,000 to $5,000 for a genome, human genome. We will be down at the $50 level in the next several years. That is going to have undoubtedly a profound impact on healthcare in times to come. So from that point of view, it's the best of times. And we'll see what that means in terms of data and integration, the kinds of other questions you were asking. In some ways, it's sort of the best of times because, you know, we have seen through NIH and other places and the private sector large increases in research budgets, in principle spurned by the human genome and other things, to actually do better at discovering drugs. But then, as you've already heard, I'm sure that you'll continue to hear throughout this, the success rate is abysmal. And you know, so this is another sort of illustration of the best of times and the worst of times. So putting that into words, you know, we have this explosion of data, which in principle allows us to do a lot. We are starting to get, and I'm going to emphasize this, we're starting to get to an understanding of complex systems in ways that we never have before. That should be impacting the drug discovery process. At this point in time, I don't think it is, but I think we're on the cusp of a turnover, and I'll say more about that. And of course, the information technology that's needed to do all this, which is an area where I spend quite a lot of time myself, is obviously improving. So let's optimism rule. So what this all means is, you know, this whole traditional model that's been based on, in the computational side at least, computational chemistry and cheminformatics, we're now bringing the idea of bioinformatics and systems biology, studying the whole system, particularly in silico, into play into this whole process. And the end result of that is something I would call systems pharmacology. And it's actually doing pharmacology in a different way than we've done before, which I'll illustrate. And I'll show you a couple of examples of what I mean by that and why I think it's so important. All right, so what's my agenda? I'll give you a little sense of, you know, I think what you're going to get out of this course is, because this is the nature of the game right now, you're going to get from different people, you're going to get some mixed messages. Their opinions are going to be somewhat different about where drug discovery and the whole industry is going. So I'm just giving you my perspective. I'm going to tell you where it comes from, and you can you can take it or leave it. That's the business. I'll say a little about the omics revolution in a bit more detail, something that I'm very passionate about, and something that I think is also driving some change. And anything that drives change, I love. So the notion of open science, and I mentioned the IT revolution, but open science is also something that's changing. Drug companies are becoming, at some levels, much more open than they ever were before. And that's basically out of necessity because of failure, my opinion. Again, this is bias coming in, right? And then what impact does all that have on drug discovery? We'll look at that, and I'll give you a couple of examples, because how could I possibly stand here and not talk about my own work? People who work, of course, I don't actually do anything, but the work of people in the lab. And then there's a few words of caution about everything I've said, which probably means it's not true at all. All right, so this is, what we do is I maintain a resource, an open resource called the Protein Data Bank, which is the repository for all macromolecular structure information, proteins, DNA, RNA. And there hasn't been a drug discovered in the last 30 years that hasn't used that quite extensively. And that's given me a certain viewpoint in all of this. We distribute a ton of data. There are 7 structures looked at every second, essentially, around the world. And then we've been using that ourselves in a research perspective to do what I've already introduced to you, the notion of systems pharmacology. So, and personally, I mentioned that open science. Also, I've started some companies while I've been here, which is one of the reasons why I'm doing this Associate Vice Chancellor job right now, because you'll learn as you go on, the more you complain about something, if you complain long enough, before you know it, you're part of the solution. So trying to form companies here is not totally straightforward. So finally the Chancellor says, well, you don't like it, fix it. So that's kind of what I'm trying to do right now. So that's sort of some background. So let's look at the omics driver. So right now, we're in a very interesting situation associated with data, particularly genomic data, but it's true of all data in biology and beyond, for that matter. We're completely out of whack with the ability to handle that data. So the data, you all know what Moore's law is? Yeah, most of you. So Moore's law is just a reflection of the dropping cost per some unit of computing over time. And essentially what you could do with a unit of computing, whether it be storage or CPU, was essentially that doubles every 18 months. And that it's gone between 12 and 24 months, but 18 is a sort of average. And biological data of all ilks, and the whole notion of translation and having data not just at the omics level but at higher levels, all of that data for a long time that was essentially keeping track with paralleling Moore's law. So the cost of maintaining that data was essentially on par, hadn't changed over time. That is no longer true. In fact, with 4th generation sequencing, the newer types of sequencing technologies, the amount of data far, far outweighs. This is an exponential scale, and you can see, sequencing sort of was paralleling this for quite a while, and then, first of all, with 2nd and 3rd and 4th generation, that's really gone completely wacky. Right now, the amount of sequence data that we're generating is doubling every 5 months. It's absolutely huge, and that doesn't speak to any of the datas of greater complexity, which I'll get to in a minute. No one is really addressing the issues of what we keep, what we throw away. We're not addressing issues associated with the complexity of that data, the errors in that data, how we represent metadata, how we represent provenance, which is how we describe the value of that data and who owns it. All of these things are completely out of whack right now.